after studying the details of mitosis and meiosis, you are now able to recognize that there are similarities and there's also differences between these two processes. I mean, they're both dealing with cell division. They both have events that relate to the nucleus and to the chromosomes and how chromosomes are separated into groups. And we have these stages called the prophase, metaphase, anaphase, telophase that are repeating between these two processes as well. But there's also going to be many differences, and that is what I want to focus on in this presentation. Let's recall, for example, when we talk about mitosis, that this type of cell division is going to be one that uh, mitosis is going to be a division that takes place in the cells of your body, the cells that make you function and your metabolism and your organs and organ systems. Those are the cells that have been produced by mitosis. We call those cells somatic cells, and they have two sets of chromosomes. When mitosis happens, we know that the original cell, the original parent cell, is going to divide, go through those steps you learned about, prophase, prometaphase, uh, metaphase, which you see here with the chromosomes, each consisting of two chromatids, the original chromatid plus the identical copy or sister chromatid, and they line up on a region called the metaphase plate before the chromatids, the sister chromatids, are split in anaphase, and eventually the cell uh, completes dividing its nucleus through telophase, and then the rest of the cell, cytoplasm and plasma membrane, are going to divide through cytokinesis. By the way, remember that telophase and cytokinesis refer to different things. Telophase is division of the nucleus, cytokinesis is division of the cytoplasm, but they happen simultaneously. They're happening at about the same time. And that mitosis is going to end up producing two cells. What is different between mitosis and meiosis is that in mitosis, only one round of division is necessary. The cell divides just once into two cells, and that's it. It's the end of the process. Meiosis, however, is going to have two rounds of division. Let's take a look at those two right from the beginning. The cells that participate in meiosis are special cells designated as germ cells. These germ cells exist only in the reproductive organs of organisms that reproduce sexually. In the case of humans, these germ cells will be housed in the testes of men and in the ovaries of women. So, those germ cells are also diploid. They have two sets of chromosomes when they begin the process of meiosis. Before meiosis, there's going to be an interphase where each chromatid makes a duplicate, the sister chromatid, and that is what you see here in this illustration. Again, for the sake of simplicity, we see four duplicated chromosomes. If you see in this illustration, those are the single unduplicated chromosomes, just one chromatid in each chromosome. But here we're looking at four duplicated chromosomes. And so what happens in meiosis is that in the prophase one, there's going to be that synapses. There's going to be the union of homologous chromosomes. Homologous chromosomes are those that have the same size, the same shape, they have the same locations for specific genes in those chromosomes. We call that genetic loci. They have the same genetic loci. And after they join in synapses, then they're going to exchange corresponding matching pieces of chromatids in a process called crossing over. Crossing over does not happen in mitosis. Homologous chromosomes in mitosis, they ignore each other, there's no interaction between them, but homologous chromosomes, they do engage in synapses and crossing over in prophase one. Then later, what we see in metaphase one, which is different also from the metaphase of mitosis, is that the homologous chromosomes are still attached. And so they are lining up the metaphase plate as tetrads. Homologous chromosomes in synapses are called tetrads. And so tetrads are aligning in the metaphase plate. But over here in mitosis, you saw that it was just the individual duplicated chromosomes that are lining up along the metaphase plate. And then what happens? Anaphase 1 is going to separate the two homologous chromosomes into separate cells. And watch what happens here. This is really important. When anaphase happens and the subsequent telophase 1, the nuclei that are produced are going to now be haploid. Remember, I just mentioned that we begin meiosis with the cell that is diploid. It has two sets of chromosomes. But now that we end up with telophase 1, the nucleus made in telophase 1 in each of the cells is going to have only half the number of chromosomes we began with. I mean, take a look at the illustration. There's only 1, 2 chromosomes, but we began with a cell that had 1, 2, 3, 4 chromosomes. So the reduction in chromosomes that is accomplished through meiosis happens right there during meiosis 1. At the end of meiosis 1, the cells you have are going to be haploid already. But division is not done yet. And I want you to understand the following. Division cannot end here because each chromosome still consists of the two chromatids, sister chromatids, that are still attached. So some people explain it this way. The reason we have to do meiosis, again, meiosis 2, is because we have to split those sister chromatids apart. And that is exactly what would happen in meiosis 2. Meiosis 2 has the prophase 2, metaphase 2, anaphase 2, telophase 2, and the purpose is now to produce cells where there's going to be just a single chromosome made up of a single chromatid because sister chromatids have been separated. Because each of the two cells made at the end of meiosis 1, each divides and makes two cells. In the end, at the end of meiosis, you're going to have 4 cells, and each one of those 4 cells is going to be haploid. They will contain only half the number of chromosomes that we saw in the original germ cell that began the process of meiosis. So that's, some of the things that you need to keep in mind in terms of comparing mitosis and meiosis. In your textbook, you're also going to find another useful table, and you can look at this and confirm some of the information we have described before. DNA replication before mitosis happens during the S stage of interphase. Cells that go through meiosis, germ cells, also have an interphase, and it's during the interphase that sister chromatids are going to be made. That's the only time when DNA is replicated. What is DNA replication? DNA replication is when a chromosome is duplicated, a copy is made of it, and you end up with the two sister chromatids. Okay, so both of these events, mitosis and meiosis, have an interface with DNA replication that precedes them. Number of divisions? Now you know it. In mitosis, there's going to be only 1 division. In meiosis, there is going to be 2 divisions. Remember, with meiosis, you need to add the suffix. If you're talking about prophase 1, we're talking about the first prophase during meiosis 1. If you're talking about prophase 2, that's the second prophase, the one that happens in meiosis 2. So remember to add the suffix. If I ask you, for example, hey, when does crossing over happen? Don't just tell me prophase. You have to add the prophase 1 because it's during meiosis 1. If I ask you, when does independent assortment happen? You must say it's during metaphase 1 because it's during the first round of meiosis when it happens. Don't just say metaphase. All right, so the suffix is important. What else? Synapsis never happens in mitosis. We said that. Synapsis does happen in meiosis, and it's going to be one of those events that contributes to genetic recombination and genetic diversity for species that reproduce sexually. Then in terms of the number of daughter cells, the daughter cells that are made in mitosis, here's a few details to remember. It's 2. They're going to be genetically identical to each other and also genetically identical to the parent cell. Not only that, those 2 daughter cells, like the parent cell, are going to be diploid. in the case of meiosis, we're going to see 4 daughter cells that are made. But those 4 daughter cells are going to be genetically different from one another. Why? Can you think? Yes, you remember we had prophase 1 with crossing over, metaphase 1 with independent assortment, and because of those events, the 4 daughter cells are going to be genetically different. And they're also different from the parent cell because the daughter cells that are made at the end of meiosis are only haploid. But the original parent cell, beginning meiosis, the original germ cell was diploid. So all these cells are going to be different. In the end, the purpose for the cells made in mitosis is going to be for growth, for development, for repair after an injury, and in some organisms, mitosis will provide the extra cells needed to make a new organism by the process of asexual reproduction. Asexual reproduction is what plants can do. For example, when you have the clipping of a root or a stem, or sometimes a leaf of a plant, and from that root or stem or leaf, a new plant develops. That is asexual reproduction because no sex cells were needed for the new organism to be made. In the case of meiosis, the cells that are made are going to continue the process of maturing and sometimes developing further into the gametes, the sex cells that will be used for sexual reproduction. And remember, in addition to sexual reproduction, the cells made in meiosis also contribute to the genetic diversity of a population, which gives the population a greater chance of surviving if there is a change in the environment or there is a disease that breaks out within the members of the population. That's the details about mitosis and meiosis. I want you to compare and I want you to remember.